So we're going to watch a little bit of this awesome McGraw-Hill animation, cellular respiration, today, and I'm going to just point out to you a few things that are important and worth noticing here. So I'm going to go ahead and play it, and then I'll pause it when I want to point things out to you.
The process of digestion results with carbohydrates and other molecules being removed from the consumed food and transported into the bloodstream. From there, nutrients, like the carbohydrate glucose, will leave the bloodstream through a capillary wall and enter a tissue cell.
So notice that the glucose started in the digestive system. That's what we called stage 1 of metabolism. And then the blood transported it into the cell, and now we're going to have stages 2, 3, and 4 happen inside of the cell.
Once inside the cell, cellular respiration will completely oxidize the glucose molecule.
Also notice this video focuses on glucose, but this could be happening with amino acids and could be happening with fat products, glycerols or fatty acids. We're just taking a look at glucose. And one other thing to look at on the structure here, each of these black balls, these represent a carbon. So glucose is C6, and so we're going to watch those 6 carbons as this continues.
Releasing high-energy electrons. The overall goal is to make ATP, a storage form of energy for most cells. Cellular respiration is a 4-stage process that begins with glycolysis. Glycolysis literally means splitting sugars, and it is the first step of cellular respiration, occurring in the cytoplasm of the cell.
Catch that? Glycolysis happens in the cytoplasm of the cell.
Glycolysis consists of 2 distinct phases, an energy investment phase and an energy harvesting phase. In the energy investment phase, 2 ATP molecules transfer energy to the glucose molecule.
So notice those were ATPs that came in and then they left as ADPs here. So these are phosphate groups that just got added on to glucose, and we saw those on paper when we did the glycolysis pathway. So this is kind of the particle version of what we did on paper.
Forming a 6-carbon sugar diphosphate molecule. This molecule splits, and the energy harvesting phase begins.
So we saw the energy investment was putting the phosphates on, and now we've got energy harvesting, or production, where we're going to get energy back out.
During this phase, the 2 3-carbon molecules are converted to pyruvate, and ATP is formed.
Notice it took 2 ADPs initially during investment, and then I'm getting 4 ATPs out now, so that's a net of 2 ATPs, right? If it cost me 2, but I gained 4.
Glycolysis is a 10-step reaction that involves the activity of multiple enzymes and enzyme assistance. In the process, a net of 2 molecules of ATP, 2 molecules of pyruvate, and 2 high-energy electron-carrying molecules of NADH are produced.
So a couple things. Remember, it was glycolysis, so we had 6 carbons, and now we're down to 2 3-carbon chunks, so that's lysing or cutting one of these glucose molecules here. Something else on the NADHs, these are what we call high-energy coenzymes, right? And these little stars here are going to represent the hydrogens, and they have an electron with them. So those are going to be ultimately the energy that we want out of our food. This is not the major source. Glycolysis is not the major source of NADH, but we'll see that'll come into play later in the electron transport chain.
When oxygen is present, the pyruvate molecules and NADH enter the mitochondria, and the next stage of cellular respiration begins.
So notice here, these particles now are going to go all the way to the inside of the mitochondria. And just to give you a second picture of that, we've moved all the way into the matrix, the dark green of the mitochondria here. So this is where the citric acid cycle is going to be happening. So all the way in here in the matrix of the mitochondria.
The next stage of cellular respiration involves the movement of pyruvate into the mitochondria, where it undergoes oxidation. Each pyruvate molecule is converted into a compound called acetyl-CoA. In the process of pyruvate oxidation, electrons are transferred to NAD, producing NADH, and a carbon is lost, forming carbon dioxide.
So remember we had 2 3-carbon pyruvates initially, and now the 3-carbon pyruvates are down to 2-carbon acetyl groups. These are my acetyl groups right here. So each 3-carbon pyruvate has lost a carbon here. Oh no. Okay, let's get back to where we were.
The next stage is the citric acid cycle, also called the Krebs cycle. Here, acetyl-CoA will bind with a starting compound called oxaloacetate, and through a series of enzymatic redox reactions, all.
So this is going quickly here, but let me back up so we catch that. So these are the 2 carbons from your food, and these are 4 carbons that are going to basically stay home in the citric acid cycle over and over and over again. But these are your food carbons right here that are being metabolized.
Acetate. And through a series of enzymatic redox reactions, all carbons, hydrogens, and oxygens in pyruvate ultimately end up as carbon dioxide and water. The pathway is called a cycle because oxaloacetate is the starting and ending compound of the pathway. For every glucose that enters...
So let me pause you a sec. You see how we have our 6 carbons here, and then we're going to lose one of those carbons and another one. What you want to pay attention to in the citric acid cycle is that we lose that carbon dioxide, and now basically all that's left of those food molecules are these high energy hydrogens with their electrons, and they are attached to these high energy coenzymes, NADH, we get 3 of them, and then 1 FADH2. What the video goes on to talk about is if I had a glucose, then this would actually run through twice, and the narrator rattles off all kinds of numbers. You don't need to memorize or know those numbers. You just need to know if one acetyl group comes in, what do I get out of this? Be able to interpret your pictures like you see in the workbook. The reason we just focus on one acetyl-CoA is because you could get acetyl groups from not just glucose, but you could get them from a fatty acid, you could get them from an amino acid, so it makes it more generic to just think about this happening once. So I'm going to skip through all of her second cycle, and now we're going to look at the electron transport chain and oxidative phosphorylation. So we've been doing the citric acid cycle in the matrix here. Now we're going to see that these next enzymes are embedded in the membrane here. So again, big picture, now we're going to be looking right where it says ATP synthase. These reactions are going to happen across this membrane. We're going to pump hydrogens to the light green area, and we're going to have the coenzymes from the citric acid cycle starting in the dark green area. So we just produced them there, and then they're going to come and react with these enzymes here. One other thing, as you see those enzymes show up, you can look at these enzyme structures on MolView, some of them if you want to. So for example, here is, let me shrink, oh, can't shrink this down, but here's a quick glimpse of the cytochrome C oxidase. And so remember when we see these cute little enzyme shapes that they're actually these massive ribbon diagrams or these massive proteins. All right, so let's go back and see what happens in our final pathway.
The electron transport chain is a series of membrane-bound carriers in the mitochondria that pass electrons from one to another.
And that big protein I just showed you is this cytochrome oxidase here. So this is not actually just a solid blob, it's a large protein, large membrane protein.
As the electrons are transferred between the membrane proteins, the cell is able to capture energy and use it to produce ATP molecules. Proteins in the chain pump hydrogen ions across a membrane.
So do you see how the NADH came up, and the little bright spots, those are my electrons, and then the H+, the protons, move into this intermembrane space. So my H that comes in is basically 2 pieces. It's an electron that's going to move through, and it's an H+ that gets pumped across the membrane here. As we continue.
When the hydrogen ions flow back across the membrane through an ATP synthase complex, ATP is synthesized by the enzyme ATP synthase.
So what she just described is that this is what's called an electrochemical gradient. It's similar to what we have with sodium and potassium pumps, where we force those protons over here. They want to move back across. We have the same thing in a battery. Those Ions say, you know what, they're too concentrated here. They want to move back across. And so as those protons move back through ATP synthase here, they provide energy, and that energy is what enables us to take our ADP and turn it into ATP. So this is called an electrochemical gradient. We have a disproportionate separation of charges or electrons on chemicals. So an electrochemical gradient drives this ATP synthase.
Oxygen acts as the terminal electron acceptor. By accepting electrons, oxygen is reduced to form water.
So here's the oxygen. This is these two oxygens, and these are the electrons coming in, and so oxygen is going to accept those electrons. Over here we would have seen that the coenzymes became oxidized, and so here's the other half of that, that the oxygen becomes reduced. It picks up those electrons.
A byproduct of the electron transport chain. All the high-energy electron carriers from the previous stages of cellular respiration bring their electrons into the chain. From this, the bulk of ATP from the entirety of cellular respiration is produced.
So she's about to say that there's, I think, 32 ATPs here. What I want you to focus on is, yes, we saw ATPs in other portions of this pathway, but we get a much, much larger quantity of ATPs produced during the electron transport chain and the oxidative phosphorylation that's happening here. This is where the vast majority of ATP is produced. So if anything goes wrong with one of these, like cyanide, for example, binds to, I believe it's complex 4 here, that gets you all the way through the pathway. If this is broken because cyanide is there, you can't produce ATP and your cells will quickly die.
Net of 32 to 36 ATP. In summary, we have seen how the 4 stages of cellular respiration are responsible for converting the energy found in the glucose molecule into ATP.
So just a final observation here. We're inside of the cell here. This is a membrane channel that's allowing glucose into the cell. Here's glycolysis in the cytoplasm with a net of 2 ATP. Inside of the mitochondria is where, if there's oxygen present, aerobic reactions happen where we have pyruvate get oxidized. Citric acid cycle is inside of the matrix here, produces a little ATP, but not a huge amount. And these numbers would be for a glucose molecule. And then the electron transport chain is present in this inner membrane of the mitochondria. And you can see how much bigger the numbers are here. This is where we're going to get the bulk of the ATP. So I hope that kind of stopping and commentary was helpful to you. Sorry it took so long, but hopefully that helps you. And as you rewatch the video on your own, it all makes a little bit more sense.